Hello and welcome to my YouTube channel, my remote class. Very good evening to everyone, and today I shall take class on proteins. Proteins are one of the most important biomacromolecules in our body. Proteins are present in all the cells. There is not a single cell which has without protein. Proteins plays a very vital role in cellular functions. For example, it helps in growth and maintenance. It helps in repairing our body tissues, and all the enzymes are proteins in nature, and they regulate all the biochemical activities or the metabolic processes. There are lots of hormones which are proteins in nature, and they act as messengers. Proteins also help in producing immunoglobulins so that they can fight against all the infection. Keratin, elastin, collagen, they all play a very vital role. So let's start with the structure of proteins. So what are proteins? Proteins are actually organic compounds which are made up of carbon, hydrogen, oxygen, nitrogen and sulfur. And there may be phosphorus, iron and other elements. So what are proteins? Proteins are organic compounds which have carbon, hydrogen, oxygen, nitrogen and sulfur, and there may be phosphorus, iron and other elements also. Or what are the structural units of protein? The structural units are amino acids. Now before going through the details of the structures of protein, let us go through some of the general properties of proteins. Number one, they are insoluble in water. That means if you try to dissolve proteins in water, it won't get dissolved. Second point, they are all colloid in nature. We have already discussed about colloids in our previous session on carbohydrates. And thirdly, the pure proteins are tasteless. Fourthly, they are colorless. These are regarding pure proteins. Fifthly, if you give heat, then proteins will be coagulated. You have seen this thing in our daily life when we prepare omelette, that albumin part gets coagulated and we see a whitish thing. Now if you provide X-ray or ultraviolet ray or alcohol or detergent, then what will happen? Or say acetone, then what will happen? The 3D structure of protein will get denatured. That means the geometric shape will be just destroyed. Proteins can be precipitated by magnesium sulfate or sodium sulfate or ammonium sulfate. So these are the things which can precipitate proteins. And finally, proteins can change color in biurate solutions or in Million's reagent. So these are all the general properties of protein. Now let's go through the structure of protein. Now the proteins may be of four types. One is called primary proteins. The second one is called the secondary proteins. Third one is called the tertiary proteins. And the fourth one is known as quaternary proteins. So let's go through number 1, number 2, number 3 and number 4 respectively. All the amino acids are present on a straight line and they are linearly arranged. Here you can see all these are amino acids all these units are amino acids, and they are all on a straight line and they are all linearly present. For example, glycine. This is an example of primary protein. Now the secondary proteins are of two types. One is called alpha helix. Another one is known as beta pleated sheet. Let's draw the structure of each one. For example, in case of alpha helix, it looks like this. This is alpha helix. What about this beta pleated sheet? It looks like this one. That means like the wavy roof of asbestos, like the wavy rooftop of asbestos. Third one is known as tertiary structure. It looks like this. That means it has a 3D structure, quaternary structure. Now what are quaternary structure? In case of quaternary structure, two or more identical or different polypeptide chains present together. So it will look like this. So this is a quaternary protein structure. That means in case of quaternary structure, we are getting more than one amino acids are present. Now let's see the structure of amino acid. How does it look? Amino acid are the structural units of proteins, as I said before. And how do they look? They look like this. Now this is the structure of amino acid, where R varies in each amino acids. There are 20 amino acids in our body, and this R varies in each of them. So if we look at the structure, we find that this part is known as the amino part. This part is known as carboxyl part. Now if you look at the structure, then you find that C, or the carbon, is present at the center, and there are 4 different groups in 4 corners. Here is H, here is NH2, here is R, and here is COOH. This is known as the Juiter ion, and this carbon is known as alpha carbon. Alpha carbon is nothing but the carbon which is attached adjacent to the carboxyl group. Now the value of this R varies. For example, if R equal to H, then we call it a glycine. If R equal to CH3, then we call it alanine. If R equal to C3H7, we call it valine, and likewise. So this R group varies in each amino acids. Now as I said before, that the proteins are made up of amino acids. For example, let me draw this picture. Now for example, this is a group of children. This is a group of children, and these are individual child A, B, C, D, E. Now look at this picture, you will find that they are all holding hands together. They are all holding hands together. And likewise, The amino acids also holding hands together. These hands are known as the peptide bond. This hand is known as peptide bond. If there are two peptides, then we call it dipeptide. If there are more than two, we call it polypeptide. Poly means many. So let's see how does it form. So how does a peptide bond form? So let's draw two amino acids, and then we have to add another amino acids. These OH and H will react together to form a water molecule. So next structure will look like this one. So what will be the peptide bond? The peptide bond will be this one. This is the peptide bond. Amino acids are of three types on the basis of reactions, of course. It may be neutral, it may be acidic, and it may be basic. Now what happens in case of neutral? There may be the same number of amino groups equal to carboxyl groups. That means the positive and negative here are equal. That means it is neutral. What happens in case of acidic amino acids? More carboxyl groups, less amino groups. These are acidic amino acids. And what are basic amino acids? More amino groups, less carboxyl groups. So what are the examples of this one? For example, in case of neutral amino acids, it may be valine, it may be leucine, for example aspartic acid, or maybe glutamic acid. The examples of basic amino acids may be lysine or arginine. There may be another way of classifying amino acids. It can be classified into essential amino acids and non-essential amino acids. Now what are these essential amino acids? The essential amino acids are not synthesized in our body, but we require these amino acids daily. We require these amino acids daily. So what we have to do? We have to take from, and non-essential amino acids are produced in our body, so they are not essential. Now let's go through the examples of essential amino acids. Threonine, lysine, methionine, arginine, valine, phenylalanine, leucine, isoleucine, tryptophan, and histidine. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. These are all essential amino acids. Now on the basis of presence and absence of essential amino acids, the proteins may be of two types. One is called first class protein, where all essential amino acids are present. Where all essential amino acids are present. And what are second class proteins? Where all essential amino acids are not present. Now all the animal proteins are first class proteins. All the animal proteins, whereas all the plant proteins are second class proteins. Now let's go through the classification of proteins. Now the proteins can be classified into simple proteins, conjugated proteins, and derived proteins. First one is simple proteins. Now the simple proteins is made up of only amino acids. They may be again classified into two groups. One is called globular, another one is called fibrous. So simple proteins are made up of only amino acids, and they can be classified into two categories. One is called globular proteins, another one is called fibrous proteins. Now what are globular proteins? Where the shape of the proteins are globular. They are spherical in shape. For example, globulin. And if the proteins are thread-like, hair-like structures, slender, thin, slender, fiber-like structures, then we called it fibrous proteins. The example is globulin, and here the example is keratin or collagen, or maybe fibrin. Second one is known as conjugated protein. Now what are conjugated proteins? Where the protein is associated with a non-protein thing. That means proteins must be present, and with them there is another thing which is non-proteins. So proteins are associated with non-protein things. For example, nucleoprotein. Here we find that there is protein, with it there is nucleic acid. Then another example, glycoprotein. There is protein, and with it there is glycogen. Then we have another example, lipoprotein. There is protein, and with which lipid is present. Finally, we have derived proteins. Now derived proteins are those proteins which undergo hydrolysis, or coagulation, and a new protein is formed. So what happens? A protein undergoes hydrolysis or coagulation, and as a result what happens? A new protein is formed. For example, trypsinogen, then fibrinogen, then peptone, then peptides. They are all examples of derived proteins. For example, trypsinogen gives rise to trypsin, fibrinogen gives rise to fibrin, and these peptides and peptones are formed during the digestion of proteins. I hope you have understood today's class, and if you have any query or any problem, then do comment in the comment section box. I shall definitely reply. And if you like my video, then do like and share. Thank you for watching.